So today we'll talk about CNS. We'll for sure get through this PowerPoint. It's not as long. And then PNS is a little bit lengthy, so we might split it up into two. Okay, but, so for central nervous system, we know from yesterday, going over just basics, okay, that it involves the brain and the spinal cord. Okay, so those two structures anatomically create the central nervous system. Okay, now, when we're developing, okay, we develop from what's called a tube, a neural tube. So we start off as a tube, which is kind of odd, but really, truly miraculous how it turns into all of the brain structures that we're identifying in lab. So what I want you guys to know is I want you to know the primary brain vesicles, of which we have three: the pros, the mes, and the rhombencephalon. Okay, PMR. Pros is in the front, mes is middle, rhomb is in the back. Okay. Now those primary brain vesicles, those primary enlargements, ultimately become adult brain structures, okay, with the ventricles or the spaces that are associated with them that we've already looked at in lab. What you don't need to know, what you can cross out, is secondary brain vesicles. I'm not going to ask you that. That's neuroanatomy. Those of you that take it in grad school, you'll get there, don't worry. Okay, so for now, secondary brain vesicles, we don't need to go to that level of detail. It's not an embryology class. Okay, so from the forebrain then, we develop the cerebrum, which includes our hemispheres that we've identified, and our what's called the diencephalon, which I'm going to repeat again today. I've got another slide that lists this. And the diencephalon has two structures that we've identified, the thalamus and hypothalamus in lab, and then we're going to look at the epithalamus today, which is where you find that pineal gland. It's just a separate region, but the epithalamus is essentially where the pineal gland is. And since the cerebral hemispheres come from this prosencephalon, we've got lateral ventricles that go along with it. The thalamus and the hypothalamus have the third ventricle that ultimately goes along with it as a space that houses fluid, okay, inside those regions. So for the mesencephalon in the middle, we're going to get the midbrain, which is the beginning portion of the brain stem. We'll talk about that in a little bit. And that's where the cerebral aqueduct is located. Okay. So then finally, most posterior, we've got the rhombencephalon, and that becomes the remainder of the brainstem, which is the pons and the medulla oblongata, and then we throw the cerebellum in there too, just posterior to it, and that's where we find the fourth ventricle. Okay. Okay. So that's it for the level of detail on this that I want you to know. Again, most of these structures nicely we've seen in lab, so we have a general idea of where they're located in the brain. Okay, so the way we're going to structure the central nervous system in terms of lecture and information is we're going to cover 4 different areas, okay? Actually, you can add a number 5 because we're going to do the spinal cord too. We'll get to that. So first we're going to talk about the cerebrum, which is the main real estate. Okay, the cerebral hemispheres have the main real estate of the brain. It also includes the lobes, okay, the frontal lobe, parietal lobe, temporal lobes, occipital lobes, okay. Second, we're going to get into the diencephalon, which includes the thalamus and the hypothalamus. Okay, we've identified that in lab. Interestingly enough, the thalamus, three-dimensionally, is a very bulbous structure. It's got like 2 bulbs on it, one on either side, and it's got these big hooks that kind of come off of it. Okay, on those sagittal brain models, you just see it as a circle. You can't really appreciate the actual shape of it. And then the epithalamus is going to be in the back here, right above the superior colliculus. It's like hidden, and that's where that pineal gland is that you guys identified on the sheep brain. Okay, so that's the diencephalon. So third, then we'll talk about the brainstem, which includes 3 parts. The midbrain is kind of where we saw those cerebral peduncles when we were looking at the sheep brain. So the cerebral peduncles are going to be where the midbrain is. And then the pons is below that, and most inferior is the medulla oblongata, which then leads us to posterior to these guys is going to be the cerebellum. So we'll talk about the cerebellum and its function. And then number 5, we'll talk about the spinal cord. Because again, CNS is brain and spinal cord. Okay, so we're going to talk about brain and spinal cord. Okay. All right, so as far as the CNS goes, all right, the way the gray matter and the white matter is organized is slightly different, okay? So let's review. What type of structures do we find in gray matter? Just rattle them off. Interneurons, what else?
Dendrites, axons.
Okay, axons of interneurons. What other specific structures? I can't hear you. Horns? Horns are composed of gray matter, but what physical structures do we find in gray matter? Right, so the whole function of gray matter we decided was what? Analysis and integration. So what type of structures do you have to have there in order for analysis and integration to occur? I'll give you a hint. Synapses, okay? So synapses involve what structures of the neuron?
Axon terminals.
Axon terminals of one and
Dendrites.
dendrites of another. In addition to cell bodies, but dendrites of another. Right. Okay, so we find all of those structures in the gray matter, which leads to analysis and integration. Okay, good. And then we decided that the white matter is composed mainly of myelin, which is what makes it white, but specifically what type of structures do we find in white matter that are myelinated? Axons, right? So we decided that the whole function of white matter is what?
Transmission.
Transport and communication, right? Communication between higher processing centers. Okay, so transport and communication of signals. Okay, so we've looked at this picture at the bottom here before. Okay, spinal cord, we've looked at this in lab. We know that the gray matter of the spinal cord is deep to the white matter. It's got this butterfly type effect to it. Okay, in the middle there's a central canal or a central cavity that's filled with cerebrospinal fluid. Okay, but the brain's organization is a little bit different, okay? So the brain has deep areas of gray matter, okay? Yes, there are deep areas of gray matter, okay? But specifically, if we're talking about the cerebrum, okay? So this is cerebellum, here's cerebrum. Typically in the cerebrum, we have an outer layer of gray matter. Okay, we have an outer superficial layer of gray matter, and that's called the cerebral cortex. Okay, you find those in both cerebral hemispheres. We'll talk a little bit more about it, but that is your conscious mind. Okay, so up here, synapses up here allow you to be aware of what's going on. The fact that you're awake, the fact that you're driving, the fact that you're dressing, the fact that you're studying. Consciousness occurs when synapses fire in this cerebral cortex. So this little couple millimeter thick layer in both cerebral hemispheres is what allows you to interact with the world and know where you are and what you're doing. Okay? Okay, so you're only gonna find that in the cerebrum, okay? Another structure you're gonna find in both the cerebrum and the cerebellum are these deep clusters of gray matter, okay? A little bit different than the spinal cord. So you're gonna find deep clusters of gray matter. And we're going to call those, in general, okay, we're gonna give them a name just because it's easier if we identify them as something. We're gonna call those brain nuclei. Or you might see the word basal. Both start with a B, okay? Nuclei. And those nuclei we can define as collections of cell bodies in the CNS. Okay, because we didn't identify what clusters of cell bodies in the CNS are. Clusters of cell bodies in the PNS are what? Ganglia. So we've already defined that. So now we're defining the clusters of cell bodies in the CNS. And we're going to call those, in general, brain nuclei. There are different names depending on where you are in the brain. We are just not going there. Okay, there's just way too much information. We're trimming it down and we're summarizing. This is just intro. Okay? Okay. All right, so there's more gray matter in the brain than there is in the spinal cord, because the brain has more processing ability than the spinal cord does. Okay? So if we had to compare gray matter between the spinal cord and the brain wins. Okay? There's more analysis and integration that happens in the brain than the spinal cord. And it's because of this extra layer of cerebral cortex that gives us our conscious mind. Okay? Okay. All right, so let's just, again, some of these structures nicely you've already seen in lab, so it's a little bit of a review. So for the cerebrum, again, we know 2 cerebral hemispheres, a left and a right. Okay? We have an outer layer of cortex. So on this cadaver brain, you see this darker outer layer here, that's the cortex. And then deep to that is white matter. Okay? The corpus callosum we know is kind of the union between, it allows for communication between the 2 hemispheres, which is nice. And then we know that a gyrus is basically a bump and a sulcus is a depression. Okay? And the longitudinal fissure is what separates the 2 hemispheres. So nothing new here. We've looked at this already in lab, which is nice. Okay. So the cortex, cerebral cortex. So we're only talking about the couple millimeter thick gray matter that's on the outside layer of the cerebral hemispheres. Okay, that's all we're talking about here. This is your conscious mind. Okay, so we're going to have, because it's gray matter, we're going to have cell bodies there, we're going to have dendrites, we're going to have axon terminals, you're going to have unmyelinated fibers, some interneurons in there, okay, because it's gray matter. All right? So this cortex is split into functional regions, sensory and motor regions, and we'll talk about where in the cortex these different regions are. Okay, so motor obviously is going to control motion, voluntary motion, and sensory is going to give us our conscious awareness of sensation. What type of sensation do we have? And then in the brain we have other areas that I'll show you called association areas, and they allow us to give context to what it is we're experiencing. Okay? So I'm feeling temperature. Okay, great, but is it hot or is it cold? And am I associating it with too hot or too cold or not hot enough or not cold enough, kind of the variation that you can have in your sensation? The association areas allow you context so that you can place whether it's appropriate or not. Okay? Okay, so let's talk about the lobes. We've looked at these in lab. Okay, they're located just below the skull bones that live right on top of them. So frontal, parietals, temporals, posteriors, occipitals. Okay, and the one lobe we didn't talk about, but we need to be aware of, is called the insula. And the insula is a small lobe. It's not like a pretty round lobe. It's just a region that you have to basically pull the temporal lobe kind of away a little bit in order to see it. It's deep. It's deep and medial to the temporal lobes on both sides, okay? And that lobe is called the insula, all right? So let's talk about each lobe and what it's responsible for, all right? What are the responsibilities? So the frontal lobe. In lab, you guys had to identify the central sulcus. And you'll note that on those brain models, it's not a perfect line that you can identify. You can kind of guesstimate where it would be, okay? And the central sulcus depression essentially separates the primary motor cortex of the brain from the primary somatosensory cortex of the brain. Okay, so it's just a nice landmark, okay? So the frontal lobe, okay, is where motor information is processed. Okay, so your primary motor cortex is in the frontal lobe. This precentral gyrus, so this big bump, okay, precentral gyrus because it's before the central sulcus, it's called a precentral gyrus. That is purely motor. And that's where pyramidal cells are going to synapse. We'll talk about crossing over and stuff later. But those pyramidal cells that we looked at in lab, they're going to synapse there, and they're going to provide some sort of motor response. Okay, so they're synapsing in the frontal lobe. specifically in the primary motor cortex. Now, other areas in the frontal lobe, okay, include this number 45 and 44 in the picture, dotted area called Broca's area. You have that on either side of the brain. So you have a left and a right, okay? On the left side, so the left hemisphere, Broca's area, okay, is the physical production of speech. Okay, because we're in the motor cortex, we're talking about motor. So this is your physical ability to voluntarily contract your masseter, your orbicularis oris, your zygomaticus major, all the muscles that you need to contract in order to produce speech. Okay, that comes from this area on the left-hand side, Broca's area. On the right-hand side, Broca's area provides emotional overtones to your speech. Okay, are you happy? Are you sad? Are you angry? Okay, it's going to give context or emotion to what it is that's coming out of your mouth. Okay, so this kind of lends itself to where people say, oh, I'm more left brain than right brain. You know, people tend to say, oh, if you're more left brain, you're more scientific because it's more set in stone. And if you're right brain, you're more emotional. You base your decisions more on your emotions. So if you hear that, that's where that's coming from. Okay, the premotor cortex is basically just the cortex in the front, a majority of the frontal lobe, and that's just responsible for higher level processing. Again, we're not going to go into that level of detail. There's so much information. We're just sticking to the basics. So frontal, prefrontal cortex, primarily motor, pyramidal cells are going to synapse here, and we have Broca's area that's responsible for the physical production of speech on the left, and then on the right, you've got that emotion that you're adding to it. Okay? That's where we're going to stick it. Okay, the homunculus. Homunculus is basically little man, means little man. And it's basically a map that scientists developed, and they took a specimen, a human, and stimulated different parts of the cerebral cortex, both motor and sensory. Okay? And for motor, they basically looked to see what contracted, what moved on the person's body. For sensory, they basically asked the person, where do you feel this? Okay, so they developed this map of the body based on stimulation of the cerebral cortex, both sensory and motor. And what it really just tells you is how many nerves and nerve endings are devoted to different areas of the body, okay? So your face, both sensory and motor, very fine motor skills, right? To be able to talk, blink, facial expression, okay? That takes a lot of neural information. So that's gonna take a huge part of the map, right? Same thing with sensory. Your face is very sensitive. Your lips, your eyes, your nose, okay? Contributing to those special senses. So that's going to take a large real estate of the sensory map. Your tongue, sensory and motor. Okay, you've got fine movement of your tongue in order to produce speech. And then your tongue is also responsible for taste, right? Getting whatever it is you put in your mouth back to where your taste buds are, which can fire off neurons that synapse in your sensory cortex to tell you what it is that you're tasting. Is it sweet? Is it sour? Is it salty? Is it umami? Like, what are we doing? Okay? So the tongue has big real estate. What about your hands, right? Super fine motor control, super sensitive. All right, you get a paper cut, you feel it, right? Ouch, right? Just a simple little paper cut and you feel it. So again, it's just showing you how sensitive different parts of the body are. Your feet too. Not only are they responsible for your natural gait, your walking, okay, but you need to be able to feel your feet in your shoes, in your socks, right? And if you don't, then that's going to affect your walking. So things like that, okay? So all I want you really to appreciate is that there's a sensory map and a motor map to the body. And there are certain parts of the body that require more nervous innervation and are more sensitive than others, okay? Contralateral means that one side of the body is in charge of the other side, or one side of the brain is in charge of the other side of the body. So essentially, your left side, your left cerebral hemisphere, is responsible for what you're experiencing on the right side of your body. Your right cerebral hemisphere is responsible for what you're experiencing on your left side of the body. Okay, and I'll talk about that more. So that's what contralateral means. Again, we covered that term on day one, which seems so long ago. Okay. Okay, so we talked about motor cortex. Let's talk about the sensory cortex. So sensory information takes on different lobes. Okay, so we're going to talk about each one. And the sensation that we're covering is just basic sensation. Touch, ability to see, ability to hear, smell, your special senses, as well as somatic senses like vibration, tickle, temperature, pain, all of that good stuff. Okay? All right, so let's start with the parietal lobe. So parietal lobe, okay, is going to be part of that primary, it's basically the post-central gyrus. The post-central gyrus is called the primary somatosensory cortex. Okay, so that's where you're getting conscious awareness of your somatic senses. Okay, and we define somatic as what? A body region not in a body cavity. And we gave 3 examples of the body that are somatic. What are they? Skin, bone, and skeletal muscle. Okay, so those sensations from those areas are coming from your parietal lobe, specifically the primary somatosensory cortex. Contralateral meaning, again, the sensations that you're experiencing on the left side of the body, your right parietal lobe is responsible for that. Okay, the sensations you're getting on the right side of the body, your left parietal lobe is responsible for that. Okay. Okay, so just posterior to the primary somatosensory cortex is that association area that I told you about, which gives you context. What are you sensing? Okay, are you sensing pain? Are you sensing temperature? Are you sensing vibration? Okay, so the association area takes the fact that yes, you're sensing something, but what? So again, further analysis of what's going on. The brain is so complex. I mean, to just sit and think about it is just wow. Like you just can't even begin to imagine. And how fast it is. How fast you can sense what you're doing at any nanosecond. It's just amazing. Okay. Okay. So temporal lobes on the side. Temporal lobes is where we get our hearing. This is where our primary auditory cortex is. We have an association area that gives context to what is it we're hearing. Okay, are we hearing somebody screaming? Are we hearing a fire engine? Is somebody honking behind you because you're looking at your phone at a stoplight? Like what's the context? Okay. And in the temporal lobe, again, outlined here, okay, is another area. This is called Wernicke's area. Okay, there's a left side and a right side. The left side Wernicke's area allows you to understand what someone is saying to you. Okay, so yeah, you can physically talk, but you've got to be able to understand that, you know, letters come together to form words, words come together to form sentences, sentences have meaning. Okay, I mean the level of complexity just goes up and up and up. So left side Wernicke's area allows you to understand spoken word. And then right side allows you to understand the emotion that's coming from that. Okay? Okay. So if somebody, for example, had a stroke, or say they had a brain injury, and Wernicke's area was affected, okay, would they still be able to speak?
Yes.
Right. Would it make sense?
No.
No. So they'd be talking gobbledygook to you, but it would make perfect sense to them. And you'd just be like, okay, yeah, like, oh boy, yeah. Okay. Versus somebody that has a brain injury to Broca's area, okay, would they be able to physically talk?
No.
No. But would they be able to understand what you're saying?
Yeah.
Yeah. So these are people that probably would use a whiteboard. if they had the physical ability, you know, they didn't lose any other kind of issue as part of the brain injury, but they might use a whiteboard to communicate with you, because they can't get it out. But they know what they wanna say, and it makes sense. So I mean, if I had a choice, I'd rather have an injury to Broca's area, because I wouldn't wanna live life, you know, thinking that what I'm saying makes sense, and people just give you a blank stare, you know, like that's gonna be really hard, because then you're kind of stuck within yourself, and there's no way of getting out of that, okay? Okay, all right, so then the insula. Pull away the temporal lobe deep inside. All I want you to know, just get rid of equilibrium here. I want you to know that your sense of taste comes from the insula. Okay, so the fact that you have umami and you guys have foodies and you love going to certain places and you love enjoying food, which is part of living, we have our insula to thank for that. Because if we didn't have an insula, or all of our connections to the insula were damaged, doesn't matter what you put in your mouth, you wouldn't know what it is. You wouldn't be able to enjoy it. Okay. All right. Okay, so that leaves us with the occipital lobe. This is where we get vision. Okay, this is visual cortex. Don't worry about the calcarine sulcus, that's just a landmark that kind of shows you where it is. So there's a visual cortex, which is occipital lobe, and there's an association located within there. So again, it gives you context. What are you seeing? Are you seeing rain outside on a rainy day? Are you experiencing an earthquake and you're seeing, you know, your pool swish back and forth? I mean, there's so many different experiences, right? The occipital lobe allows you to have. So that's where our visual conscious ability comes from, is the occipital lobe. Okay. All right, so these deep gray matter, okay, in the cerebrum, I said that we're gonna call them, in general, basal nuclei or brain nuclei, clusters of cell bodies in the CNS, okay? What are they responsible for? So they're gonna receive information, because it's gray matter, so there's gonna be analysis, they're gonna receive sensory information, and they're gonna decide, okay, is there gonna be, what kind of motor output are we talking about? But these brain nuclei are involved more so in intensity of movement, okay? So not the fact that you're physically moving, that's the frontal lobe's job. But now the deep clusters of gray matter are responsible for how intense do those movements need to be, right? Like, are you running a 100-yard dash where you've gotta like throw it all into like 5th gear? Or are you just simply using your stylus to take notes and lecture? Like, totally different. So your brain nuclei helps control that based on the situation. And it also allows you to pick the proper muscles to use, right? Because you don't want to have full body muscle contraction all the time, you wouldn't go anywhere, right? So if you're getting, for example, a gallon of milk out of the fridge, you're probably going to be using your biceps to flex the elbow and hold that gallon. But at the same time, your triceps can't be contracting, because that's gonna, you know, that's going to defeat the purpose of what it is you're trying to do, the movement that you're trying to make. Now I can go into concentric and eccentric contractions, but we're not gonna go there. So basically it's just, it tells the appropriate muscles, you're in charge, and this is what task you need to do, right? So again, the level of complexity goes up and up and up. And if you're doing something repetitive, like over and over again, like those rhythmic gymnasts, oh man, I mean, they've trained, their brain is trained, right? They got the hula hoops, they got the ball, they got the ribbon, I mean, everything they do is super rhythmic. So, I mean, their basal nuclei are on point because they've had tons of training. Okay, lots of integration going on during those. This is just, so again, this is just a transverse slice through the brain, and the purple and the pink and stuff is just showing you there's different nuclei, okay, deep clusters of gray matter in the brain, collections of cell bodies that are responsible for intensity of movements, picking the right muscles, turning off the right muscles, okay? Yes, they have different names. We're not knowing them in here, okay? That's neuroanatomy. get into grad school, trust me, it's coming. But for now, we're just gonna summarize it and call it brain nuclei, okay? That's all we're doing. Okay, all right, so moving on from the cerebrum, now we've progressed to the diencephalon, okay, which includes the thalamus, which we've identified in lab, and the thalamus is huge. The thalamus is the main sensory routing area in the brain. All sensory information that's coming from the spinal cord and other areas of the brain has to synapse in the thalamus, and the thalamus will decide where in the brain that message is going to go. Okay, so is it information about vision? Okay, the thalamus recognizes that and says, okay, you're going to the occipital lobe. Is it information about motor movement, motor responses? Okay, you're going to the frontal lobe. Is it information about hearing? Okay, well, you're going to the temporal lobe. So the thalamus is the routing station for all sensory information that's coming into the brain. Okay, so it has a huge, huge responsibility. What I just said. Okay, so the hypothalamus, just below it, okay, it's kind of like a region. We identify it's like a region, but it's kind of a big deal. Okay, when we talk about the endocrine system, we'll see why. But the hypothalamus basically tells the pituitary gland when to secrete hormones. Okay, so we've identified the pituitary gland on the brain model. It's a small little nugget, but man, it's got like 8 or more hormones that it secretes. And those hormones go into the bloodstream and they go to different areas of the body and they do different jobs. All coming out of that little pituitary gland. But the hypothalamus controls when that's secreted. And from what part of the pituitary? Because we know there's an anterior and a posterior pituitary. Okay, the anterior pituitary will make, but the posterior pituitary does not. The posterior pituitary can store it, but they're made in the anterior pituitary. So anyways, we'll talk more about that in ANS. So the hypothalamus controls the autonomic nervous system, so it's going to control and have overall dictation of sympathetic and parasympathetic nervous system, fight or flight, arrest and digest. Okay? It's involved in you regulating your body temperature, telling you whether or not you're hungry or thirsty, and regulating your circadian rhythm, so your sleep cycles. Okay? Emotional responses and memory, too general, so don't worry about those. These are the big guys. Okay? Okay. So epithalamus, again, that's that pineal gland, right? And the pineal gland secretes what hormone? Melatonin. So that also consists and helps with sleep cycles. Okay. All right. So that's pretty much the diencephalon. Okay. So we'll start with the brainstem now. So the brainstem includes, I'm just going to label it directly on this picture, the midbrain, the pons, and then the medulla oblongata. Okay. So the brainstem is basically the big connector between the brain and the spinal cord. Okay. So all neurons that are coming from the spinal cord to the brain are going to pass through the brainstem. Okay. And 10 of our 12 cranial nerves that we've already looked at in lab come off the brainstem. Okay? Everything except for olfactory and optic. Everything else comes off the brainstem. So the brainstem is going to be involved in hearing and visual reflexes, which I'm going to talk about in the next slide. So this is just kind of an intro. All right, so posteriorly behind the thalamus, we have the tectal plate, right? What's it also called? Corpora quadrigemina, which means body of 4 in Latin. And on the top we have what? Superior colliculus, and on the bottom? Inferior colliculus. Okay, so why do we have them? Superior colliculus, responsible for vision and visual reflexes. Okay? So you go to the eye doctor and he shines that light into your eyes to see your pupillary reflex. That's a reflex. You walk into a movie theater late and it's pitch black dark and you're coming from a bright sunny day directly into the theater and you can barely see. Well, that's because your pupil has to close down and get adjusted to the lighting conditions that you are now in. That's a visual reflex. Okay? Okay, inferior colliculi, hearing and auditory reflexes. Okay, someone lights off a firecracker behind you and you didn't know it was coming. You turn directly towards that side. Right? That's an auditory reflex. Okay. One area in the brainstem, specifically the midbrain, I want you to know, and it's only because when this goes south, okay, it creates a pathology. So the substantia nigra is responsible in general, okay, to control unwanted movement. So it helps you control movement, okay? So it limits excessive movement. It allows for smooth transitions in movement, okay? If this starts to head south and starts to degrade, which in people it does as you get older, you can develop Parkinson's disease. And Parkinson's disease, one of the primary symptoms is you can get tremors. And tremors are unwanted muscular movements that you have no control over. Okay, I've treated hundreds of Parkinson's patients in the clinic, okay? Unwanted muscle movements. So lots of tremors that can lead to balance problems, okay? So if you lose the ability to control unwanted movement, you're gonna get excessive movement, and it can impair your functional mobility, okay? Don't worry about the red nucleus, okay? Again, the cerebral aqueduct we've looked at in lab. Cranial nerves 3 and 4 actually come off of the midbrain. So when you guys are identifying them, if you're looking around the cerebral peduncles, that's where you're going to see cranial nerve 3, which is what nerve?
Trochlear.
3 is oculomotor, 4 is
Trochlear.
Got it. Okay. Okay, so the pons. Pons is just inferior to the midbrain. You've got cranial nerves 5, 6, 7, and 8 coming off of it. Pons is a big deal mainly because of this. It controls respiration and your bladder. Okay, those are the big pons responsibilities. Okay, there's other stuff, okay? It's involved in other stuff, okay? But these are the big ones. Okay, so cranial nerve 5 is what? Trigeminal. 6, abducens. 7,
Facial.
facial. And 8,
Auditory.
auditory or vestibulocochlear. Okay, good. All right, so the medulla, all right? At the very inferior aspect, it's got these structures that we call pyramids. I just think of pyramidal cells, synapse there. So they're gonna have pyramids, okay? And these pyramids are where axons come in, pyramidal axons, and they cross over to go to the opposite side of the brain, okay? So axons from the right side of the body, because we're looking at an anterior, sorry, this is a posterior view. This is posterior, anterior, this is posterior view. Okay, so this is left side. So axons coming up on the left are gonna cross over in the medulla oblongata and go to the right side of the brain. Okay, vice versa. Axons from the right side of the body are gonna come into the medulla, cross over to the left, and go to the left side of the brain. Okay, that's contralateral. But the medulla oblongata is huge because blood pressure and heart rate are controlled. Blood pressure, heart rate, and respiratory rate. Okay, pons has some respiratory properties. Okay, medulla controls the rate. All right, so if you have a brain injury in the medulla oblongata, okay, survival is not likely because it's just too important. It helps regulate blood pressure, heart rate, and respiratory rate. Okay. All right, cerebellum. So cerebellum is most important with posture and equilibrium. Okay? So your sense of balance comes from the cerebellum. Okay? It also contributes and works with the basal nuclei to contribute to coordinated and smooth body movements, but that helps with posture and your equilibrium, your ability to be upright. Okay? So someone that has like a cerebellar stroke or pathology to the cerebellum will probably have very uncoordinated movements, not be able to maintain balance, be very impulsive. I've treated these types of patients before. They're just all risks waiting to happen. They just have no concept of the fact that the minute they move, they're probably gonna end up on the floor because they don't have that coordinated, smooth movement pattern that everybody takes for granted. They rush into things and they just do it and don't think about the repercussions. So these are types of patients that they may be independent and they may have really good strength, but you've got to supervise them because they're going to be super impulsive and they're going to be, a harm and a risk to themselves. Okay. All right, enough from that. Now, the coverings. We've seen this in lab, which is nice, okay? So, we know that the deepest covering that covers the brain and the spinal cord, and I said think of it as saran wrap directly on top of the organ, is called what? Pia mater, so it's light blue in this picture, okay? Then just above it, we have a purple layer here, called what? Arachnoid, or also you can call it arachnoid mater, okay? And then the most superficial layer is called what? Dura mater. And dura mater actually has 2 layers to it, okay? Which I'll talk about in a minute. So in between the arachnoid layer and the pia mater is a space. What's that called? The subarachnoid space, yeah, named for exactly where it's located, and that's where you find vasculature. That's where you find blood vessels coming in to feed and bathe the brain as an organ. Okay. But also, okay, we need to realize that in this subarachnoid space is where cerebral spinal fluid flows, which we call CSF. So you not only have blood vessels that are, doing passive transport, bringing nutrients and getting rid of waste to the brain itself as an organ, but you've got cerebral spinal fluid flowing through the subarachnoid space. And if it's a fluid in your body, what's it a derivative of? Your bloodstream. So how convenient. You've got blood vessels right here, and cerebral spinal fluid is going to be flowing through the subarachnoid space. Okay? Now, the subarachnoid space and the arachnoid mater kind of make these little involutions, and they're called arachnoid villi is plural, or villus, okay, is singular. It's kind of like an outcropping. It's like a papilla. It's like a bump kind of a thing, okay? And these villi specifically situate themselves near these big structures that we call sinuses. Now we've heard of sinuses before when we talked about the heart. What sinus did I mention? The coronary sinus. Now sinuses aren't strict direct blood vessels, but they do carry blood and they deliver it to places depending on where it needs to go, okay? So we have sinuses also in the brain that are designed to pick up fluid, resorb it, and bring it back into the bloodstream, okay? And the one example here is called the superior sagittal sinus, which we will get to next week when we do human vessels in lab. We'll identify the superior sagittal sinus. I'll show you on the next slide as well. Now, the dura mater, okay, the most superficial layer, has 2, okay, layers. The most superficial layer is called periosteal layer because it attaches to the periosteum of bone. Deep to that is the meningeal layer because it contacts the meningeal layer of arachnoid, okay? Now granted, there's a small space in between there called the subdural space that we identified, okay? But it's called meningeal because it's closest to the meninge, that is arachnoid. So both of these layers separate and create these sinuses. So that's why the dura mater has 2 layers, because the layers split and create these structures called sinuses that are designed to collect fluid from the brain and deliver it back into circulation. Yeah, I'll talk more about that in a minute. Okay. All right, so bucket handle head guy that you guys looked at yesterday. Okay, you identified the falx cerebri, the falx cerebelli, and the tentorium cerebelli. Those are involutions of dura mater that are designed to protect the cerebrum and the cerebellum. Okay, the brain's not just wishy-washy in your skull, it is situated such that it is not going to move. And it has multiple layers of protection, including the 3 meninges and the falx cerebri, the falx cerebelli, and the tentorium cerebelli to protect them. Okay, so within the falx cerebri, we have a structure called the superior sagittal sinus. Superior because it's on top, sagittal because it's going in what plane? Sagittal plane, sinus, because it's a structure that's holding fluid. Okay. Yes, we have an inferior sagittal sinus at the bottom. We have a transverse sinus in the transverse plane. We'll identify these again next week in lab when we do special circulation. Okay, so these are designed to drain blood and fluid from the brain, enters your jugular veins, goes down towards superior vena cava back to the heart. Okay. Okay, so ventricles. Ventricles we've identified in lab. We know they are spaces in the brain that house fluid, specifically cerebrospinal fluid, which is a derivative of your bloodstream. Okay. All right, so we know the lateral ventricles live in each hemisphere. That's why we have a left and a right. Okay. They're connected by the interventricular foramen, which then connects to what ventricle? The third ventricle, okay, which lives near the thalamus and the hypothalamus, right, the diencephalon. And then you've got the cerebral aqueduct that connects the third ventricle to what? The fourth ventricle, right? And then the fourth ventricle ultimately connects to the central canal of the spinal cord, where cerebral spinal fluid can flow all the way down the length of the spinal cord, okay? Now, ventricles is where fluid flows, okay? But that fluid is made by a special structure called the choroid plexus. We don't have that to identify in lab. You can actually see it on the models. We really should add it. But it's called the choroid plexus, and you only find that structure in the ventricles. And the choroid plexus is what makes cerebral spinal fluid. So it's going to take what it needs from the bloodstream and create cerebral spinal fluid. Okay? So this bottom picture here is kind of just a little snippet of a choroid plexus, okay? And that structure is going to locate itself near, what a big surprise, a capillary, okay? Because that's where goodies can enter and exit, okay? And on the outside, so the whole perimeter here is the inside of a ventricle. And it's lined with, here we go, ependymal cells, which we said circulate cerebrospinal fluid. They have cilia and they circulate the CSF. So choroid plexuses are basically taking blood, taking whatever ions and goodies they need from your bloodstream, and creating cerebrospinal fluid, which is then flowing through your ventricles, and the ependymal cells are making sure that that happens, flowing. Okay. Okay, so this is kind of like a summary slide to show you the flow of cerebrospinal fluid. So up here in pink, okay, we have our choroid plexuses, and the arrows are just showing you that cerebrospinal fluid is being made. Okay, it's being made. We have choroid plexuses down here in the fourth ventricle. Okay, we have it in the third ventricle, all the ventricles. Okay. So that fluid, that CSF, is going to flow through the ventricles. But when it hits the fourth ventricle and goes into the central canal, okay, which it will, it also has to have an area to enter the subarachnoid space. because it needs to bathe the brain itself, okay? Not just the ventricles inside, but it needs to bathe the brain itself. And the way it does that is through the subarachnoid space. So we're not gonna name the apertures, okay? Just think of the apertures as doorways. And that's doorways for CSF to leave the ventricles and enter the subarachnoid space, where it can bathe the cerebellum, okay? It can bathe the cerebrum, and then all of a sudden in the cerebrum, because we said CSF flows through subarachnoid space, we have those little outcroppings called arachnoid villi. Well, the arachnoid villi allow us to resorb that fluid and take that fluid through a sinus of some sort and bring that back into the venous system via your jugular veins, which are taking it back to the heart through superior vena cava. Okay? So the brain does not need a lymphatic system to drain its fluid. Okay? It uses this technique. It uses arachnoid villi. So arachnoid villi are going to take that fluid and throw it into the sinuses. Because again, if cerebrospinal fluid is a direct derivative of your bloodstream, choroid plexuses are constantly making it. So how do you not just, increase pressure within the brain cavity and just, pop? Well, you can't really pop because you got your skull bones, but how does that not cause increased pressure, which ultimately would damage your brain, right? Well, that's because we're getting this resorption happening constantly. So it's constantly being resorbed back into the venous system. And the rate at which CSF is being made is the same rate as which it's being resorbed, okay? So that allows for equilibrium and homeostasis between the volume of fluid that's flowing. So if someone, for example, has a brain injury, right, say fell off their e-bike, which is so common now, probably took a topple forward, maybe not wearing a helmet, okay, brain injury, boom. Maybe there's a subdural hematoma or something, like a bruise on the brain. What that does is that causes swelling. It causes swelling of your brain as an organ in response to the injury. That excess swelling may not be able to be handled by the arachnoid villi and your sinuses. It's too much, okay, all at once. All of a sudden you've got this giant hematoma. So what needs to happen? Well, you're in the ER, they notice you have a hematoma, and what they do is they open up, they drill through your skull, and they open up a piece of it. And they allow your brain to swell. And they allow time to give your brain and associated structures to recover from the injury. So most people, if they're in the hospital with an acute brain injury and they have a portion of their skull open, they usually wear a helmet to physically protect that area. Because you don't want, again, it's an open exposed area, so you don't want infection, number one. Okay, but you also don't want someone to like bump into it or anything to bump into it, so they usually wear like a hard hat. Okay. Okay, questions about cerebrospinal fluid flowing? I pretty much just have a summary here for you guys. Okay, so let's just review a little stuff. So some of this, again, I can just kind of breeze through because we've already seen this in lab. So let's turn to the spinal column and the spinal cord. We already know from lab, 8 cervical nerves, 12 thoracic, 5 lumbar, 5 sacral, 1 costal. On either side, we have 31 pairs, okay? 31 pairs of nerves. We know the conus medullaris, cauda equina. We know the dorsal median sulcus and the anterior median fissure. Cervical and lumbar enlargements, okay, are essentially just enlargements of the spinal cord where nerves are going to exit into your extremities. So you have a cervical enlargement because that's where nerves are entering your arms through your brachial plexus, and then lumbar enlargement where nerves are entering your legs from your lumbar and sacral plexus. Okay. Okay, so that's the only new term there, is the enlargements. So gross anatomy in the spinal cord, again, we've seen most of this. Everything on here we've seen in class, okay? We know now contralateral means a crossing over, which is also called decussation, okay? Ipsilateral means side, same side, contralateral opposite side. So the only thing that's new here are rami. We haven't talked about rami, okay? We've talked about roots, right? We've talked about dorsal and ventral roots, and I said that roots allow what to connect to what? Allow the spinal nerve to connect to the spinal cord, okay? Right, it just routes the information. Because the dorsal root carries what kind of information? Sensory, and the ventral? Motor. Okay. So, rami, if we're talking about primary rami are defined as the first branches off of the spinal nerve. Okay, so that's what we'll call it. The first branches off of the spinal nerve, not the roots. The first branches off the spinal nerve are called primary rami. And we'll really get into rami in the autonomic nervous system, but for now, we're just gonna take a back seat. Okay, so that's what rami, it's the only new thing here. Okay, all right, so again, beautiful picture, we've seen this before. Now we know that we have two different kinds of sensory neurons, and we have two different kinds of motor neurons, whether we're talking about somatic or visceral, okay? Now, what I want you to know is I want you to know where the sensory neurons terminate and where the cell bodies of the motor neurons are located, okay? So, in a nutshell, the sensory neurons, both somatic and visceral, okay, blue and green, structurally, what kind of neurons are these?
Unipolar.
Unipolar, okay? They are going to terminate in what part of the spinal cord?
Gray matter.
Gray matter specifically where? The posterior horn, okay? So in the posterior horn is where we have the axon terminals of somatic sensory and visceral sensory neurons. That's why the posterior gray horn of the spinal cord, I told you, makes a connection to the outside of the spinal cord because it's receiving sensory information. So sensory information terminates there. It's going to synapse with interneurons, okay? Interneurons in the posterior horn. And interneurons are going to figure it out, okay? There's going to be ones that are going to send information up through tracts to higher processing centers so we have conscious awareness of what it is that's going on. But as far as motor neurons go, okay, cell bodies, long axon, axon terminals, structurally these are what? Multipolar, okay? So, somatic motor neurons are red. Those cell bodies are located where in the gray matter?
Interior horn.
In the anterior horn. So you're going to find the cell bodies of somatic motor neurons in the anterior horn of gray matter of the spinal cord. Okay? Visceral motor neurons, you're going to find their cell bodies where?
Lateral.
In the lateral horn of gray matter of the spinal cord. So that's why we have different horns in the spinal cord. Different structures are located in the different horns. Posterior horn is purely sensory. Okay? Lateral horn handles visceral motor neurons. Anterior horn, somatic motor neurons. Okay, so I like this picture because it really gives you a nice colored visual of what you find where. Okay. All right. All I really wanted to just an FYI, if you know of any patients that get a lumbar puncture, they usually do it in between L4, L5, so they can get into the central canal and do the least damage to the cauda equina, because there's the most space for them to do it. So that's just like an FYI, you don't have to memorize that. Okay, almost done. So, the next three slides. Summary slides, I'm not gonna put these on the exam, I don't want you to memorize them, okay? It's just designed for you to understand the general concept of how the brain processes information, the central nervous system, okay? Now, this is showing you different pathways, actually 3 different pathways. Are we knowing the names of the pathways? No, we're not. Okay, if you take a neuroanatomy, you certainly will. But, okay, example, we know that sensory information is coming in from the periphery in some way, shape, or form. And it's gonna arouse some sort of sensation, okay? So if this is a proprioceptor, okay, this is sensing your muscle's position in space. Okay, so that's the stimulus for this pathway. The stimulus for this pathway is somebody flipping a feather on the bottom of your foot, so like tickle, okay? This one is someone lighting a match under your foot, so pain, okay? So it essentially shows you that sensory information takes it in, okay, through a unipolar neuron, right? Depending on the pathway, you may get interneurons that are gonna take that information up to specialized areas of the brain that allow you to consciously perceive what it is you're sensing, okay? And only when the synapses go up here in your cerebral cortex do you physically know what is going on, okay? So, for example, touch receptor, okay, feather under the foot. You've got higher level communication between interneurons that's going up. You get a crossing over, okay, you get a crossing over at the medulla oblongata, okay, and it comes up and it synapses where? In the thalamus, which is the main sensory relay station of the brain. Thalamus figures out what it is, okay, this is some sort of sensory information, tickle, so it's gonna send it to your parietal lobe, primary somatosensory cortex. Because it's your skin that's being stimulated, right? Only when it hits that, boom, do you realize, ooh, there's a feather causing tickle. And the sensation I am experiencing right now is tickle. Okay? Same thing with match. You got incoming information, it's gonna cross over, and depending on the pathway, it can cross over soon, it can cross over later, it just, but it's always gonna synapse in the thalamus. And the thalamus is gonna tell it where to go. Okay, so this is gonna be more of like a frontal lobe synapse, because most likely you're gonna have some sort of motor response, which is to get your foot the hell away from the match. Right? So if you think about how many pathways we have, and how many experiences they're responsible for, it just like blows your mind, you know, no pun intended, as to the level of complexity and detail and the speed at which this happens, right? So from here, all I want you to realize is that tracks that go up, okay, are carrying sensory information, okay, for analysis. Tracks that go down are generating a motor response. And tracks, again, are what? Collections of axons in the CNS, okay? So these motor pathways just look like they're all terminating on skeletal muscle. And the overall effect at these muscles is gonna be what? In general, contraction, right? Yeah. Does each one of these have a name, these pathways? Yes. Are we learning them? No. Okay, I can tell you in PT school, I had to know all the pathways. Neuroanatomy was probably one of the most difficult courses I've ever taken in my life, and it was summer intensive. Great. So a quiz, just to give you an example, a quiz in neuroanatomy was something like this. So the professor would put up something like this, no labels on it, but just the tracks, no labels. So you had to know based on where it was going, where it crossed, whether it was a sensory or motor tract, and then which tract it was, right? But not only that, he would take his little like expo mark. Again, this is just a repeat, and this is just really simplifying it. You get a stimulus, it comes up, your brain considers, analyzes it, interneurons, and then boom, is gonna generate some sort of motor response to it. And so now, when we return back to reflexes next week, it'll make more sense, because now we've talked about tracts, we talked about neurons, we talked about different types of neurons, we talked about all the terminology. Now when we talk about reflexes, it's like, okay, that's the simplest pathway in the nervous system is a reflex. That's why reflexes are so fast. You get instant results from